Hi, I'm Dale. I look after H3D's Malaria Biology Lab. Welcome to the operation. Here at Malaria Biology, our primary role is to make sure that all of the new drugs which are produced upper GCT chemistry are actually active against the human malaria parasite. In addition to that, my team, who you can see in the background, also look after selectivity, which is an assay that we run to make sure that the compounds are active only against the pathogen that they're supposed to be intended for and are not generally toxic to mammalian cells, which is what something like bleach would do. This is an important part of testing to make sure that our compounds are actually safe. Cytotoxicity assay is running this morning. We'll be looking at about 30 different compounds to evaluate their selectivity. As you can see, that assay is a very manual process. For the malaria work, we tend to use this guy here. This is a little robot made by Opentrons out of New York, and it's able to help us with the assay to add drugs, add reagents, add blood, and add parasites, and do everything we would need it to do to see what the drugs look like against the parasite. If you like, we can go take a look at the results. After 3 days in the incubator, we can get a result to see what the compounds look like against the parasite. So this is what an assay plate looks like. And of course, this doesn't tell you very much exactly as to what the result is or what's been going on in the plate. So here's a visual representation. So what we have in this plate are 3 different drugs, each starting at a very high concentration and diluted down to a very low concentration. So we get an idea of where activity against the parasite may lie. But of course, you can't see anything until you develop the plate, and we get a result like that. And what we're interested in is exactly where the color changes. The further to the left it is, the more active the compound is. Now, I can tell visually what this looks like, but that wouldn't be very scientific. A far better bet would be to put it into this machine here and get an actual numerical result. The plates are taken from the stacker into the spectrophotometer. The optical density is read. It gives us a range of numbers which we can then use to calculate the accurate IC50. That data is then fed back to UCT chemistry and is integral for the next round of design and synthesis of the next round of compound.